In part one of this video, I wanted to go over the first part of chapter 3, cell structures and their functions. And so to learn a little bit more about biology and eventually anatomy and physiology, we're going to look at what's going on inside of the cell. And so to do that, here's a few vocabulary terms for you: organelle, cytoplasm, and cell membrane. These are basically the three main parts of a cell, and then we're going to look later in the chapter into individual sections of the cell. But to begin with, organelles, these are just small organs that are inside of the cell that complete some type of function. And then you also have a liquid-based or a jelly-like substance inside of the cell known as the cytoplasm. And then surrounding the cell, you have a cell membrane, which we'll look at in just a few minutes. A lot of times you see the cell membrane also called the plasma membrane. This diagram, this picture, figure 3.1, gives you a good outline of just a general cell. Many cells have different shapes, but this one is kind of rounded, and it shows a lot of the different cell organelles, such as the mitochondria, and then little small ribosomes that are there, lysosomes. We'll talk about these in a little later. And then you can see the cell membrane on the outside. So to kind of begin our discussion of what a cell is, we need to kind of look at some of the functions. Basically, a cell is defined as the smallest unit of life. This is the smallest living portion of the organism. It's also where the cell metabolic processes take place, and energy is created and used within the cell. It's also in charge of synthesizing or producing, creating molecules. It has some communication functions as well, and that is through various receptors and signals and chemicals. And it also has the ability to reproduce and to divide, go through cell division, and also pass along the genetic information through the inheritance. So as we kind of take a tour of the cell, let's start on the outside. We'll kind of work our way inside. On the outside, you do have a cell membrane, also known as a plasma membrane. And basically, you have just a little vocabulary here for you. Make sure you realize intracellular means anything on the inside of the cell. So that would be where the organelles would be, and the cytoplasm would be intracellular. And then anything outside of the cell would be referred to as extracellular. It's also good to realize that the cell membrane is a barrier, but many times it is a selective barrier, which means that allows certain things to pass through the cell membrane and other things are kept out of that cell membrane. We'll see examples of that kind of throughout the chapter and throughout the course as well. Now, the current model for that cell membrane is what's known as the fluid mosaic model. There's a diagram I'll show you in just a minute that kind of diagrams what that cell membrane looks like. It's basically made out of different types of organic molecules, the first being phospholipids. And these are lipids that have basically create a water insoluble function to it. So it only allows certain molecules to pass through. If they are lipid soluble, they pass through this membrane. That membrane also contains cholesterol that helps to kind of stabilize that phospholipid layer that's there. There's some proteins embedded in the membrane. Many of those are transport proteins or receptor proteins. We'll see that later. And then also some carbohydrates that are there. Notice that the phospholipid comes in 2 layers. So you'll have 2 layers of a phospholipid membrane for all the membranes of the cells. And that's what's called a bilayer. So it has 2 layers. So in figure 3.2a, you kind of see a general picture of a cell membrane and what's known as the fluid mosaic. Mosaic meaning you've got multiple types of molecules kind of throughout the membrane. So for example, again, you have carbohydrates, some lipids, some proteins, but it's also known as fluid. In other words, some of these molecules can kind of move around. They're not static, they're more dynamic, and they can move around within the membrane. Now, one topic that's discussed here is moving things in and out of that membrane, because within that cell, remember, you have intracellular on the inside, extracellular on the outside, and so you have water and ions, different molecules that can move past inside and outside the cell, and so that allows for transport. Well, you have two different types of transport. One is referred to as passive, one refers to as active. The main difference here is that passive doesn't require any kind of energy to transport a molecule across the membrane, whereas active transport does require energy, and that form of energy is known as ATP. And so we'll talk about some examples, different examples of each of these. This lists those examples for you. We'll talk about these as we go through the slides. The first three up here, diffusion, osmosis, facilitated diffusion, these are all examples of passive membrane transport. So none of these require energy. They take place automatically. Diffusion, osmosis, facilitated diffusion do not require energy. The second set of these, active transport, secondary active transport, endocytosis, exocytosis, these all require the ATP energy to take place. So first of all, let's look at diffusion. Diffusion, basically, they put the definition really here at the bottom. I know there's a lot of information on this slide. I'm not going to go through it all, but basically diffusion is the movement of some molecule from a high concentration to a low concentration. And that's usually down some type of what they call a concentration gradient. So usually you have a membrane, and on one side of the membrane you have a higher concentration of that molecule, and on the other side of the membrane you have a lower concentration of that molecule, and that allows for movement to go from a high to a low concentration. Then if you're not familiar with these terms, make sure you realize what a solution is. A solution is a mixture of these two things. The solute is what gets dissolved inside of that solution, and the solvent is what's actually doing the dissolving portion. And so, for example, if you have a solution of salt water, well, the water is the solvent, the salt would be the solute, and you mix those two together and you have a solution. So in this little diagram, very simple diagram, they just show examples of diffusion inside of this beaker of water. The water would be the solvent. They've put some substance, they've added some type of salt crystal, maybe they've colored it green so you could see this, and then what will happen is, just because of diffusion moving from high to low concentration, the molecules will start to kind of move out into that solvent, into that water. Now, to help in some of the movement, you have protein channels. These are like gates or openings in the membrane. And so you have two examples here, leak channels and gated channels. Leak channels are always open, and so that allows certain ions or certain molecules to pass through, make it a little easier for those molecules to diffuse across the membrane. Gated channels work the same way, except just like the name implies, they have a gate, and they have a system or a gate that closes that channel and will only open unless in the presence of something that has to open the gate, such as a molecule has to open the gate, or a voltage, a volt has to open the gate. But so you either have a leak channel that's always open, or you have a gated channel that is can be controlled, open or closed. So this is actually a nice diagram that kind of illustrates diffusion through the membrane. Here's our lipid bilayer. You got two layers of lipids. That's our cell membrane. Here is, you can see some molecules that are lipid soluble will diffuse directly across without any problems going inside. So this is intracellular on the inside. Out here would be extracellular on the outside. Here you have some type of protein channel. Maybe it looks like a leak channel because there's no gate here. That allows molecules from the outside to pass through a little easier going through that channel. Then notice that some molecules, remember this cell membrane is selective, so some molecules can't pass through. and so it won't diffuse. These are non-lipids, so they don't diffuse very easily through the membrane, and maybe they're too large to go through this membrane channel, so they are prevented from going inside of the cell. Now this figure 3.5 tries to illustrate leak versus gated channels. And so, for example, here is a leak channel. There is no gate here, and so it's always open. And so this basically allows molecules to diffuse across. Remember, diffusion should be going from high concentration to low concentration. So the idea here is that you have ions. These happen to be potassium ions in higher concentration, right? I've got more potassium in different places here. And so it wants to travel from a high concentration of potassium out of the cell to this extracellular area, and there's only a few of those potassium ions out here. Whereas here, notice that this sodium channel is closed. It's a gated channel, but it's closed, and so that means nothing can pass through here. On the far right, you have another gated sodium channel, but this has a molecule here called a ligand that has now opened this channel. Once this is opened, the sodium ions can still go from high to low concentration, so this is still diffusion. So these would move from high concentration of sodium into the cell to an area of lower concentration of sodium. Now we come to osmosis. This is a special example of diffusion. Most everybody realizes what osmosis is. This is the diffusion or the movement of water. And of course, again, that's going across the selectively permeable membrane. And it is diffusion of water, so that same definition applies. It goes from an area where you have a higher water concentration to an area where you have a lower water concentration. You can also kind of reverse the definition a little bit, or at least talk about it in terms of the solute. Water always wants to move toward the area that has more solute concentration, so it moves toward that higher solute concentration. And that movement, because of water, that movement because of osmosis, creates a type of a pressure, and that pressure is known as osmotic pressure. And so that's actually a pressure that's pushing up against that selectively permeable membrane. That pressure allows for certain solutions to move in either direction across the cell membrane. And so that creates a situation of either a hypotonic, isotonic, or hypertonic, and we'll look at different examples of each one of these. But the osmotic pressure here depends on the difference of the solution concentrations, because if you're not going to have you usually don't have equal concentrations on both sides of a membrane. If you do, then that's referred to as isotonic. If you have a lower concentration, notice the prefix here, hypo means lower than, iso means equal, and hyper means higher than. And so we'll talk about each one of these. A hypotonic solution, hypo means lower than, so it has a lower solute concentration. So it's less solute or dissolved substance, and it's a higher water. And that's relative to the cytoplasm of the cell. And so water, of course, moves by osmosis into the cell. And so if this cell is placed into a hypotonic solution, then water is going to want to go into the cell, causing it to swell. That could be a problem because if the cell enlarges too much, that can actually break apart or rupture. That process is called lysis. Isotonic solution, or an isotonic situation, is where the concentrations on both sides of the membranes are equal. And so you have the same solute concentration inside and outside the cell. So that's really where you want your cells to be, in that isotonic state. So your cell is not going to shrink, and it's not going to swell or rupture. It's going to stay balanced. Hypertonic solution is just the opposite of hypotonic. Hyper means greater than, more than. It's in this case the cytoplasm of the cell in a hypertonic solution has a lower solute concentration and a higher water concentration than the surrounding solution. And so water here is going to move by osmosis from the cell into the hypertonic solution. Hypertonic solution means higher solute concentration in the solution. And so that's going to mean that water goes, again, toward the area where you have more solute. And so if you put a cell into a hypertonic solution, water wants to go out of the cell into the solution to follow that solute, and that's going to cause that cell to begin to shrink. That has a special word here called crenation. It's kind of hard to see in this diagram. This is trying to illustrate osmosis. You can take a look at this in your book sometime. Kind of tries to illustrate osmosis, movement of water into movement of water from a high concentration of water to a low concentration of water, or to the area where you have a higher solute concentration. This is actually a good diagram here that shows, for example, what would happen if you would take a cell. In this case, they use a red blood cell. If you would put a red blood cell into a beaker of a solution that is hypotonic, in other words, the solution has low solute concentration. If you added this regular cell, red blood cell, into this solution, then water wants to go inside. Water is wanting to go toward the area that has a higher concentration of solute that would be inside that cell. And so that's eventually going to cause that cell to rupture. If a red blood cell is placed in an isotonic solution, remember iso is equal or balanced, and so in this case you have a balanced situation. Now notice the arrows are still going in and out. Water can still pass in and out of the cell, but everything's kept at a balance. So it's an equilibrium. So the cell doesn't get any larger, it doesn't get any smaller. If that cell is placed in a hypertonic solution where the concentration of the water around it is greater or higher, then water wants to go toward the area that has a higher concentration of solute, and so water comes out of the cell, tries to go into the solution, where eventually it's going to cause that cell to just shrivel up. Now, the last couple things about transport we need to talk about. We're still talking about diffusion or passive transport. And so here's an example of, one more example of passive transport. That's facilitated diffusion. The word facilitate means help. And so in this case, you have help from a protein channel that helps in the diffusion process. So that does not require ATP to work. And of course, active transport, we've already mentioned, does require the energy molecule. And so here's your definition for facilitated diffusion. It's carrier-mediated because it requires the presence of a carrier, in this case, a protein channel. So it's carrier-mediated. It allows the movement of substances across the cell membrane. Notice that it's still from high concentration to low concentration, so that means it's still diffusion. And that also means it does not require energy to take place. So this example shows you an example of facilitated diffusion or carrier-mediated diffusion. You have a carrier here, it's a protein channel. It's still diffusion because in this case you've got some molecule, in this case glucose, going from a high concentration to a low concentration down the concentration gradient. Active transport, remember, it's active because it does require ATP. It is carrier-mediated, so in other words, it does have to have a protein channel. But in this case, you're going from a low concentration to a high concentration, which means you're really going against a concentration gradient. And a good example of an active transport model that you'll see many times in the body, in the cells, would be what's called the Sodium-Potassium Pump. We'll see this more when we get to the chapter on muscle physiology. But here, this is a good example of a Sodium-Potassium Pump. This mechanism uses active transport to pump out or to move sodium ions out of the cells. That's against the concentration gradient. And so it's trying to move sodium ions out and potassium ions allowed to come in. And so that results in a change in the concentration gradient, which will allow for a particular type of gradient across that membrane. This diagram is kind of small here on the slides. You may have to look at this more closely in your textbook. This is figure 3.9. Kind of shows you the process of how this would work. You can kind of see the lipid bilayer here. Here's your sodium-potassium pump. This is your protein pump here. Your carrier molecule here, it does require ATP to function. So sodium ions kind of go into the channel. Whenever the energy molecule attaches, that allows this protein to kind of change its shape. That allows the sodium now to come out. At the same time, potassium is starting to come in, and it just kind of flip-flops to allow the sodium to be pumped out and the potassium to be pumped in. One other type of transport that we'll talk about is what's called endocytosis and exocytosis. As the name implies, endo means coming in, and so this is a process that's bringing materials into the cell, but it uses kind of a little chamber known as a vesicle. And so you've got three examples of endocytosis. The first one is what's called receptor-mediated endocytosis. That basically just means that you have substances that will first of all bind to some kind of receptor that stimulates this process. Once that substance binds to a receptor on the cell membrane, that triggers the cell to try to create a barrier around it or a vesicle around it, and that helps to bring that material inside of the cell. I believe there's some diagrams we'll look at on that. Phagocytosis and pinocytosis. These are other examples of endocytosis. Phagocytosis is whenever a cell is trying to bring in solid substances. That prefix phagos there means cell eating. It's like the cell is actually eating something solid. So for example, a white blood cell. White blood cell is part of your immune system. It may detect some type of bacteria or a virus infected cell, and it can phagocytize. It can actually cause that particle to go inside of the cell and transport it inside of the cell into a vesicle, and that's called phagocytosis. If the particle that it's trying to engulf or to take in the cell is more liquid based, it's a liquid or an ion type molecule rather than a solid substance, then it's referred to as pinocytosis. And pinos here refers to cell drinking. So you have a cell eating and a cell drinking phase. So this is a diagram that I mentioned on receptor-mediated endocytosis. The only difference here is that you have receptors on some of the cell membranes that whenever certain molecules would bind to this, that would stimulate the process to start the process of forming a vesicle right around these molecules, and then all of those molecules will just be engulfed inside of the cell. This is still endocytosis. It just uses receptors to start the process. The opposite of endocytosis is exocytosis, and that is a process to secrete substances outside of the cell, and that's going to use vesicles as well. Those are usually referred to as secretory vesicles because you're using a vesicle to secrete molecules outside of the cell. This is commonly seen in your glands, which we'll talk about later when we get to the endocrine system. But your glands use storage vesicles, and then those vesicles will go through exocytosis, and that helps to release the hormones or the chemicals from the cell out into a duct. And so here the vesicles move to the cell membrane, they fuse, that releases those substances or materials out of the cell during this process. And they also talk about the example of digestive enzymes. Enzymes that will digest things also can be secreted out of the cell using this process. And this is kind of showing you some examples of exocytosis. This is the cell membrane. Here is the intracellular cytoplasm. Here you have the secretory vesicle, some kind of vesicle that has maybe some enzymes or chemicals in it that needs to be released from the cell. It comes up to the cell membrane, it fuses with the cell membrane, and then it opens up and it releases those substances or chemicals out into the extracellular area. That is all of part one of this video, and there'll be a second part to go through the different structures inside of the cell, the different organelles inside of the cell.